I was going to say that there are certain events in the academic life of a school or university that I really, really enjoy. This is one of them. And being able to honor a colleague as they have attained, in some senses, the ultimate level of accomplishment in the academy, and that is to be named professor, a full professor. In this case, it's my pleasure to be here with you to honor Alexis Battle, professor of biomedical engineering, with secondary appointments in computer science and the Department of Genetic Medicine at the Johns Hopkins School of Medicine. She is also the director of the Malone Center for Engineering in Healthcare and the interim co-director of the Data Science and AI Institute. She really does an enormous amount for this school and this university. Alexis's research focuses on analyzing large-scale genomic sequencing data to unlock the secrets and function of the human genome. She uses machine learning to develop statistical models to examine how genetic differences between individuals contribute to differences in health, from cellular level changes to disease outcomes. And her talk this afternoon will explore the progress and challenges of a decade of machine learning in human genomics. As her work has contributed to understanding how individual genetic differences contribute to personal health. Those in JHU's Battle Lab share the goal of using genetic and multimodal data to guide the understanding and treatment of diseases ranging from autism to cardiovascular disease. Before we hear from others, I would like to take a moment to recognize one of my predecessors, Don Giddens, for whom this inaugural professorial lecture series is named. Don Giddens was the 5th Dean of Engineering at Johns Hopkins and a professor of mechanical engineering. Now, it's my pleasure to introduce Mike Miller, the Bessie Darling Massey Professor and Director of the Department of Biomedical Engineering here at Johns Hopkins.
Thank you, Dean Ed. Thank you, Alexis. Thank you, Alexis's family, for bringing Alexis here. Thank you, collaborator from Chicago. We have others here from not only Alexis Battle, but we also have a speaker today from California who wanted to speak. So we're really excited. I think Alexis represents what is the greatest part of Johns Hopkins, essentially biomedical science, that we are leading a discovery in biomedical science because we do it together. And Alexis was recruited by computer science and subsequently part of biomedical engineering. And that's why we have Alexis Battle here today. We have representatives from computer science as well here today. Randall couldn't be here. I asked him to give the other half. Of course, then it would take an hour, because I'm going to take half an hour, Alexis, as you can imagine. No, I'm not. Okay, I just wanted to say a couple things. So it's my understanding that in 2017, you co-authored a landmark paper in Nature that demonstrated how rare genetic variants contribute to target gene expression changes across diverse and how it changes across human tissue. So that was an important paper. And you know, in the academy, we celebrate these events. So I was really excited. I went and looked it up, and I heard a lecture yesterday, and I was told that we shouldn't be speaking in acronyms so much. And your acronym was GTEx, because the rest of the world doesn't know what GTEx means. So I went to look it up. I don't have to find it on here, but you're probably going to define it. Alexis has won multiple awards, including the David and Lucy Packard Fellowship, President's Frontier Award. And I remember when Ron Daniels called me and told me that you had won the President's Award, and I was so excited. I don't get called by the President very often, except when the faculty in biomedical engineering win awards like that, and you're one of the 3 or 4. So thank you for that. I think I'm going to turn it over now, but I just want to say again that the opportunity for biomedical engineering and computer science to work together in this new era, which really Dean Schlesinger has ushered in, the idea that we have to work together as departments because the problems are just so big. On your GTEx paper, there are a lot of other people who have won presidents, other versions of awards. It's a big problem. So thank you, Alexis, for being in biomedical engineering. You contributed so many ways. The students talk about you all the time. And one of the things I most appreciate about Alexis is whenever we're talking about structural issues in the department, Alexis speaks to what the young faculty need and what our training would be. And I notice that all the time. So thank you. So with that, I'm going to turn this over to Alexis Battle, our inaugural speaker. Oh, Tim.
I'm not Alexis Battle. My name is Yoav Gilad, and I have the pleasure and privilege to introduce Alexis. And I want to start by telling you a story. When Alexis finished her PhD at Stanford, she wanted to join Jonathan Pritchard's lab as a postdoc, and Jonathan and I back then were collaborating very closely for a number of years. And I strongly advised Jonathan not to let Alexis join. Alexis already had a faculty position lined up, and she wanted to be a postdoc for 9 months. And our work is complicated and complex, and it takes a long time to figure out what we're doing. So there's no way somebody in 9 months can do anything productive. So Jonathan ignored me. Alexis joined, and boy, was I wrong. Alexis didn't just accomplish things; she really ran circles around the rest of us. In those 9 months, she led a project that connected regulatory variation across different molecular layers, from RNA to protein data. She developed a completely new joint analysis of RNA sequencing, ribosomal profiling, mass spectrometry data, and published one of the papers I'm most proud of as a co-author. And it coincidentally was published in Science. It's still widely cited, not just for the results, but also for the truly elegant and careful analysis. Alexis, during that time and a little later, started playing an important role in GTEx. I'm not going to spell it out because apparently you need to look it up. Including a lot of papers on a little specific expression, the impact of rare variants on gene regulation. And she wasn't just working on side project of GTEx. Alexis really helped set the stage for how we think about regulatory genomics today. And Alexis and I have been collaborating since then and became not just close collaborators, but also friends. And one of the reasons I keep collaborating with Alexis, because I do not collaborate with any of my other friends, is that she never stops asking questions. And she's incredibly precise, not just in how she builds models and analyzes data, but how she thinks. And she will question numbers, and she will question anything that won't make sense to her. And a lot of the time, things that make sense to you will not make sense to her. And I'll give you 2 examples. Years ago, when we were working on really one of the first time course EQTL study, we were seeing numbers of time course EQTLs that made perfect sense to me and to my group. Nothing was obviously wrong, but Alexis was worried. And she was worried enough that we had to open up everything and look at all the analysis, and we discovered a really subtle new type of batch effect in that time course EQTL study. And if you know anything about me and my work, I realize a lot of you don't, but trust me when I say that the death knell of my career would be if I ever publish a paper with a batch effect. And so Alexis saved me and saved my group and saved the integrity of that paper. More recently, we worked on another collaborative study on dynamic EQTLs. And again, the number looked fine to me and to my students. And Alexis said that some of the functional enrichment in these numbers make no sense. And so this time we didn't even argue because of the track record and just went ahead and looked at all the code, found the mistakes, corrected it. The issue is that the mistake was in all the code that analyzed all papers and all data in my lab. So she really just turned everything upside down. But again, improved things. And for that, I'm truly grateful. She just catches not just her own mistakes, but mine too. Alexis's research has consistently pushed the field forward. She's developed methods to detect context-specific genetic effects, analyze allele-specific expression in single cell, model the effects of rare variants using complex multimodal data set, and her work has opened new direction in the study of gene regulation complex traits all the time. The papers are rigorous, useful, and enduring. They don't just make a splash, they become reference points in the field. Alexis is, as you heard, recognized with major awards. She's a Searle Scholar, Microsoft Investigator Fellow, and winner of the Johns Hopkins President Frontier Award, which is one of the university's highest honors. She received multiple Discovery Catalyst Awards. She earned all of that while quietly also mentoring a wonderful group of outstanding trainees, many of whom are now faculty on their own. But as you know, Alexis didn't actually start as a scientist in academia. She started at Google, first as a software engineer, then leading a team of 15 engineers working on machine learning and large-scale system. And I'm sure she could have stayed at Google and do just fine, but she didn't like money, so she became a scientist. But you know, every part of her science career as a PhD student, postdoc, faculty, she always got offers from places with a lot of Boston zip codes, and she never went, which I really admire, although she somehow sent all the students there. So something's going on. And as you know, she doesn't just lead from the lab. She built things here at Hopkins. She directs the Malone Center for Engineering and Healthcare, the interim co-director of the new Data Science and AI Institute. And these are real leadership roles, really shaping how data science and AI connect with medicine, biology, and with the next generation of science. What I admire most about Alexis is how she shows up. She shows up in science, in meetings, in collaboration. She's thoughtful, exacting, generous with her ideas, and truly fearless. She makes everyone around her better, including me and my group. And with that, please join me in welcoming Professor Alexis Battle.
Let me just thank Dean Ed for the introduction. Mike Miller, thank you very much. And Yoav, I finally got you to see one of my talks. I had to invite you all the way to Baltimore. But thank you for those introductions. They were very kind. So today, unfortunately, Yoav, you're still not going to see too much of my technical style of talk, because I wanted to give a talk a bit about the progress of the field that we've made in these 10 years, because it really has been kind of a wild ride. I will talk a bit about the contributions that the lab has made, but I really want to give a bit of an overview. So to begin, the overarching goal of the lab, and I see lots of my students here, is to understand individual differences. So really to understand how our individual genetic sequence, how I differ from you, how you differ from your neighbor, lead to individual differences in our cell states, gene regulation, traits, and disease risk. So this is a very broad goal, and that goal has lasted us the 11 years that I've been here, and I think will continue. We are a computational lab, and so the approach that we take is based on data and machine learning with lots of different approaches at different stages of this analysis. So connecting DNA to gene expression, inferring gene networks, connecting those DNA differences and gene expression changes to traits and disease risk. Now, I will say at the very beginning, I'm not going to draw any hard and fast lines between what I call statistics, machine learning, or AI. We've called it different things over the years. My lab uses everything from just standard hypothesis testing and linear models to probabilistic models carefully designed for our problem at hand to AI and deep neural networks. I'm not going to define these terms or draw lines between them. If you are worried about that, feel free to talk to me later. But what I am going to talk about is really the state of the field of understanding human genetic differences, some of our lab contributions, and then thoughts and lessons I've learned during these almost 11 years now running a computational genomics lab. So first, I just want to set the stage and say that this explosion that we've experienced in genomic data is enabling new science and translation that really just wasn't possible when I started. So DNA sequencing, as many of you probably know, has become very cheap and ubiquitous, powering large studies. And large studies are exactly what we need in order to look at individual differences. We need population studies across hundreds or thousands or millions of people, and that is what we're getting now with data sets like UK Biobank and the All of Us data set over hundreds of thousands to millions of individuals. But what does this really look like? So a standard study that we might look at in trait genetics is to collect genetic information genome-wide across a set of individuals for whom we also have trait information. Now, when I was doing my PhD, our studies of this form were very small, like a couple thousand people. So for height, for instance, we had like 5,000 individuals from whom we got 2 hits and a bunch of non-replicating crap. Fast forward to when I finished my PhD and was looking at starting at Hopkins, that had already increased dramatically. We're looking at hundreds of thousands of individuals. And now for height, we're looking at studies of 5 million individuals. And this is really powering us to develop things like genetic risk scores and to use it for drug target discovery that was really not possible when I was thinking about this field originally. Similarly, for other traits such as schizophrenia, where we've moved from a few thousand individuals to hundreds of thousands. And it's not just DNA. So for those studies, we're looking primarily at DNA sequence markers, but DNA sequencing doesn't just sequence DNA. We can also use it to look at gene expression, such as RNA. We can look at the 3-dimensional structure of the genome. We can use sequencing to look at epigenetics and others. So there's been an explosion not just in DNA sequence availability, but in new, very high-resolution data types that are now available not just for a few people, but are beginning to become available at this population scale that, again, we need to look at individual differences and the effects of genetics on individuals. So for example, in the space where we're looking at DNA and, for instance, gene expression in RNA, this was a big area of contribution of my PhD. But when I started looking at this, we were looking at RNA-seq data sets of like 60 individuals. My PhD thesis included an EQTL study with DNA and RNA for 1,000 individuals from just a single tissue. The GTEx projects and others started to grow this across tissues and contexts, and now we're looking at studies that have 100,000, 50,000 individuals for a single tissue, and thousands of individuals across tissues or single-cell data. So our ability to use these studies in science has grown dramatically. So this is a plot that I'd like to show just that demonstrates for trait-associated studies in red, this average sample size over years, and this really does span basically like when I was doing my PhD to just a few years ago. And what I want to point out is like, this is when I've been at Hopkins. So we've moved from a place where we had approximately 0 data to a place where we have hundreds of thousands to millions of samples available. So this, in my research statement, if you look at my application, I claimed that we had a genomic data flood, but like really it happened while I was here. And that has changed science dramatically. It's changed what I do, and it's changed what we know and what we can do with this data. Okay, so the first key result that I want to share is that we have now found just thousands and thousands of genetic loci that are associated with disease. And this has started to enable us to build good genetic risk predictors. So we can take your DNA and give you a score that tells you how likely you are, for instance, to develop schizophrenia or to develop coronary artery disease. In addition to that, we are starting, and I will say starting, to characterize biological mechanisms of these disease loci that we have found. So what does that look like? I'm going to go back to height, just as an example, again, moving from like 5,000 individuals up to 5 million individuals over the span of this time. What I'm showing here, this is not my plot, I want to be clear, this is background information, is one of the more recent GWAS meta-analyses of height that shows that polygenic risk score, so this is a genetic score, and how accurate it is at predicting a child's height compared to the accuracy of predicting that child's height based on knowing how tall their parents are, for example, or their sibling. And what we see is that very much unlike 10 years ago, genetic scores derived from these genome-wide association studies are becoming as accurate or more accurate than family history, and certainly in combination with family history are starting to show some benefit. Furthermore, when we estimate how much heritability there is from like twin studies and non-genetic data, for a long time, we were nowhere close to being able to explain all of that heritability with actual markers from your DNA. And now estimates from GWAS and others are showing that we can now account for the majority of the additive heritability of height, for example, along with other traits like this. And we've conclusively shown, in terms of understanding just the architecture, how genetics affects these traits, we've conclusively found a very long tail of thousands and thousands of small effect genetic loci in your genome that contribute to traits like height, schizophrenia, heart disease, and others, where we didn't know what that would look like 20 years ago. We didn't know if it would be 5 loci or 100, it's thousands. So we've really come a long way in understanding this. So we're building these better genetic risk scores. They're becoming more accurate for many traits. Can they be used in clinic? So I will say, this is just like to kind of understand where the field is. Evaluation of clinical use is beginning. So in this example that was published recently, they looked at coronary artery disease, and you can see that using a current score, individuals who are in the top decile, according to genetic risk, compared to the bottom decile of genetic risk, have about a 3-fold increased prevalence of the disease that they were looking at. So it looks like there's some information there that would be beneficial to a patient to know, to understand. It could guide, how aggressive you are with treatment, such as giving statins and others. But the field is very much still debating and testing kind of the difference between clinical validity, like yes, the score is giving us information, versus clinical utility. Does this really impact patient care? Can we really use this to improve outcomes? And that's still very much up in the air. Where our lab has been contributing, and this is a more recent direction for the lab, is a few different places. So we are thinking about genetic risk scores not as just an aggregate score, but can we break it down according to the component of genetic risk that's expressed through your liver, the component of genetic risk that's expressed through your brain, et cetera. So we've developed tissue-specific genetic risk scores, and these can help us find not only overall risk, but some about mechanism and perhaps key disease-relevant genes. We're furthermore working on improving their accuracy. So even though we can explain a lot of the heritability of many of these traits, our polygenic scores don't quite get up to that level of variance explained. So there's still a lot of room for improvement in the accuracy of the scores themselves. So we're working on using background information and neural net-driven approaches to improve polygenic risk scores. And we're looking at the underlying relationship between traits. So there's a lot of shared biology between the many, many traits that we've been able to look at through GWAS. And using machine learning methods, including matrix factorization, we can identify common factors that are shared across multiple traits to better understand their shared and specific mechanism. So I said in the intro to this point that we are just starting to understand disease mechanism, and I want to describe why. So most disease loci are not inside of what you would think of as a gene. So they're not turned into a protein, for example. So you would run a genome-wide association study, and most of the variants that you'll get out are what we call intergenic. And we don't know what they do just by looking at the association. So that makes it very hard to use this, for example, to develop a drug or do anything in terms of coming up with ideas for therapeutics. Our lab and others have pushed forward the use of functional data, such as QTLs, for those of you who know that term, and other uses of functional data, such as RNA and epigenetics, who left off a word there at least, to help identify what is the mechanism, what is the gene or protein that's relevant to mediating the genetic effect on the trait. However, okay, so a couple of examples of our lab working in this space. We have run large-scale GWAS meta-analyses for traits including heart failure and telomere length, and I can say from those papers that we have gone through computational characterization and experimental validation that was very labor-intensive on both sides to identify not only what are the loci that we can identify with these big, higher-powered studies, but how do they function, which genes do they affect, and can we actually demonstrate this in the lab? And that's a very laborious process. And with all of this, the field, when I say we here, I mean the field, still hasn't found a clear mechanism underlying numerous disease-associated genetic loci. So here, just to give you a definition of the term, when I say no co-localization, which is the yellow component of each bar for the traits on the x-axis, no co-localization means we can't even figure out which gene is implicated by the disease-associated genetic locus. So we don't know what most of these do. So that's kind of the state of things right now. It's very exciting developments in understanding genetic factors affecting complex traits, but we're still struggling to identify mechanism in many cases. Yeah. Great. Okay, so the next key point that I want to make, and this has been a big focus of the lab, is that genetic effects depend on context. So your DNA is not just read the same way all the time. Environment, time, and cell type really affect how your DNA is used by your cells, and thus how it affects your traits. So many different factors, we know from biology, this is not new that we understand this, many factors can modulate genetic effects, right? So your neurons in your brain read your DNA very differently than your cardiomyocytes in your heart. Whether you decided to smoke a cigarette or eat a hamburger or go for a run before you came to this lecture is impacting right now how your cells are reading your DNA. And time affects it. So this includes growth and development and aging. So this leads to lots of biological changes in your cells that can change gene regulation and therefore then affect how a DNA variant is actually going to impact downstream traits in the cell or high level. So our hypothesis when we said that we haven't found mechanism underlying lots and lots of disease risk loci, one hypothesis is that some of these genetic variants are only active in very specific environments, cell types, or time points that we simply haven't yet explored. And this paper that I cite is one joint with Yoav Gilad, who introduced me. It's a long line of research that we've been undertaking together. The first attempt to look at this in my lab was actually the GTEx project. So maybe I should go ahead and define it. Genotype Tissue Expression Project. It's a big NIH-funded consortium project looking at understanding the genetics of tissue-specific gene expression. So here we have available almost 1,000 donors with whole genome sequencing of their DNA, in addition to gene expression measured through RNA sequencing in 54 tissues across the body. Now, this is a very long line of work that I'm not going to go fully into, but I will say that from this project, we identified thousands of genetic loci that affect gene expression differently in different tissues. So we now understand how your brain uses genetic information differently from your liver, from your heart, et cetera, in much, much more detail than we did before this project. We also understand how these overlap with disease loci and additional factors like sex and ancestry. And I will point out that this data has gone well beyond our own use of it. Over 7,000 papers have now been published with the GTEx data. It's one of the most highly cited data sets available. So from this, we're able to look at context specificity. From the GTEx data, we can look at tissue specificity. And we've developed several different statistical methods for identifying context specificity from these genetic association studies. So using CAFE, for example, we can take a trait association, such as coronary artery disease, and look at all of its genetic effects, and then decompose them into which of those genetic effects appear to be active in different tissues. And you can see from this one example, but much more broadly across diseases and across tissues, we found that there's much, much more context specificity than was previously understood before we undertook this analysis. So these genetic effects that we see that are trait associated, many of them have very tissue-specific and context-specific behavior. A recent line of work in collaboration with Yoav is using in vitro cell models to very efficiently explore genetic effects across time, across cell type, and across environment. So using a system that he's termed heterogeneous differentiating cultures, we can take stem cells, iPSCs, and differentiate them into these cultures that represent multiple different cell types in aggregate in an efficient way such that we can do this across many people in a single experiment. And using single cell sequencing, we can then measure gene expression for all of these different people and all of these different cell types and perturbations on top of that to understand genetics across, again, time, cell type, and environment. So we're looking at the full course of differentiation from stem cells all the way to terminal cell types across many different people, environments, et cetera, and in this efficient system. So from this, we've seen that we do, in fact, find differentiation and time-specific genetic effects, and these genetic effects are different across different cell types. And furthermore, in some cases, these do overlap with disease loci. So what I'm showing, for example, here, if you look at the x-axis, this is essentially time from stem cell to terminal cell type. And we see genetic effects, so different colors are different genotypes across time. And you can see a big genetic effect developing only in later stages, in this case, in liver cells or hepatocytes, and basically nothing coming up for neurons or cardiomyocytes. But we see cases where there's a genetic effect in the middle of differentiation, these sort of transient effects, early stages, et cetera, all sorts of different sort of nonlinear shapes. And furthermore, we've developed computational methods that can take association studies across many cell types, across single cell data, and look for QTLs and identify when they're shared and specific to different factors. Using a method called Surge to do this type of analysis in single cell data, we've also shown that we are able to explain many more disease-associated loci using these types of analyses compared to if we just take standard EQTLs of bulk tissue data. So the key conclusions here is that genetic effects are more context dependent than we thought. Our lab with Yoav's was one of the first to demonstrate the influence of differentiation time on regulatory genetic effects. But more broadly, these effects vary across cell type, across environmental factors, and across time. And I do want to point out that analysis of these complex data really does require sophisticated computational methods. I can't just hand you the readout from this system and read it off, without doing fairly sophisticated deconvolution of the data to identify these effects. So it's data plus computation. One sort of difficult result, so talking about like where we are in the field, one thing that's remained really hard is kind of the next step. So not from DNA to RNA, but thinking about, once you have gene expression in a cell, inferring how genes work together has remained a surprisingly hard problem. This is something that I've worked on since I was an undergrad, before I went to Google. When we were working with small microarray data for RNA, we had all sorts of methods to infer gene networks in those days. And I think when I was a naive 20-year-old, I certainly would have thought that if I went from having a microarray of 50 individuals to having RNA-seq over 10,000 individuals, that I would have solved my problems in inferring gene networks. And that has not turned out to be true. So we have found a lot of useful signal in expression and splicing data, lots of work in the lab and others across the field revealing how groups of genes vary together. But inferring like the details of this network, specific edges of how genes work together or regulate each other, has just been extremely challenging. So Yoav mentioned my keen eye for batch effects and confounders. So this is something that I spend way too much of my time thinking about. But when I made this slide probably back in 2012, that's why the colors are so ugly. I made this like long list of like all the confounders that can affect RNA sequencing. And when people say batch effects, it's really not just like, well, did you run sequencing in 3 different batches? You shouldn't have done that. You can't control all of it. So there are technical variables that are different from day to day in different sequencing runs. There are biological variables that are not the one that you're looking for that are affecting all of this sort of functional omic data, RNA sequencing, epigenetics, all of that. But the result has been that confounders, and we've shown this now sort of empirically, theoretically, more empirically, that confounders really limit our ability to accurately infer gene networks. We have some recent papers demonstrating this. We also have tried learning gene networks from data covering tens of thousands of individuals, which again gives us useful signal, but it's just not super accurate at identifying the edges of these networks. So this just remains really hard. The final key direction that I want to talk about from the lab is in personal genomics. So here, we can now cheaply sequence the full genome of individual patients. So if you're thinking from a clinical perspective, it is plausible to get your genome in the clinic. And in theory, your doctors could look at this and figure out all sorts of things that they could tell you about your personal risk. It offers a hope of predicting individual disease risk. It offers a hope of diagnosing and potentially thinking of treatments for rare genetic disorders. So the promise of personal genomics seems really great, but it is really challenging. So every one of us is walking around with lots and lots of variants that are not part of any of those genetic association studies that I just described. These are rare variants that are found in very few people. You have like 50,000 of them that are at less than a 1% allele frequency, and thousands in your genome that we've never seen before. Furthermore, 5% of the population in the US approximately has a rare disease likely to be genetic. In current clinical genetics pipelines, around half of these individuals go undiagnosed. So we run exome sequencing or even whole genome sequencing, and we just can't figure out which one of these 50,000, pieces of hay is the needle that we're looking for. Like, we can't figure it out right now. So this is extremely challenging, but it does seem, this intrigued me, sort of ideally suited for machine learning approaches, right? We have to be able to make predictions based on variants we've never seen before. We can't run association studies for them. Our approach in the lab, over many years has been to build integrative machine learning approaches for personal genomics that take not only the individual's whole genome sequence, but additional data about the individual, their RNA, epigenetic data, proteomic data, and others, comparing all of this to population scale reference data, and using, in this case, a carefully designed probabilistic graphical model to take all of that information together and infer which of the variants that are most likely to be causing disruption for that individual. And we have shown that we provide much better prediction of which genetic variants are likely to be functional in that individual, and even some cases where we have been able to identify variants potentially causal for rare disease patients. So our model is called Watershed, and I just want to show you one thing here. So GAM is our genome-only model. This is like the best we can do from DNA alone. When we add in even just RNA, we improve our ability to identify which variants are causing big changes to the cell, like truly dramatically. So the best genome-only models are really kind of still very bad, and any functional data is extremely helpful. However, we are interested in what we can do from sequence alone here. So recently, there's been a lot of interest in neural network models for DNA sequence. To sort of, the information's there, it's structured sequentially like language. We've been applying, in the field, have been applying neural network models, including convolutional models and transformer models that are intended to capture like long-range dependencies in DNA to predict things like chromatin, gene expression, and other functional annotations. And this seems to hold quite a lot of promise. However, when we get to actually looking at whether these can do the job that I was just describing, really interpreting the personal genome and predicting even just what are going to be the personal changes in gene expression, we see that these models, while they do give us quite a lot of predictive accuracy, are really not very much better than standard linear models currently. They do tell us it looks like something more about potentially causal variance than otherwise. And we've seen that this is true even as the data has gotten very large. So a lot of the previous work on this has been with like 1,000 individuals. We've trained models of tens of thousands of individuals, in this case with proteomics, and seen really similar patterns. Although I will say I think that we are making progress with other ways of training these models, other objective functions, and starting to think about other architectures. So this is very much in progress. So I'll leave you with, personal genomics is an exciting possibility from whole genome sequencing. Neural networks are starting to show promise, but from our analysis, it still seems that collecting just additional data types, not even using neural networks such as RNA, protein, et cetera, is really where we're getting a lot of boost in performance here. But that will lead me to a comment just thinking about where are we using AI and genomics? Again, I don't like to draw lines between statistics, machine learning, AI that are too hard. And I will say machine learning, like sidestepping when we decided to start calling it AI again, has a long history of success in genomics. We've been doing clustering and disease classification and all sorts of things like this for years, successfully, on somewhat different problems that I'm talking about in the personal genomics space. I will also say that neural net and deep learning methods have had success, beginning in the early 2010s, really, in specific problems like variant calling, regulatory genomics, splicing prediction, and some others. So we are starting to see, some interesting applications of these methods. We've also seen cases where, they look like they might be working and they really aren't, including some that we have published in the lab, where we can see that still predicting how genes affect each other is still very hard. And even these deep learning methods, trained on lots of data, are not doing better than simple approaches. And what I've already mentioned, the case where it's still quite difficult, to predict personal gene expression. But I've been working in machine learning for a long time, and I always like to show this quote from Richard Sutton from 2019. It's his bitter lesson. And the bitter lesson, I'll let you read it, but I'll paraphrase it, is that computation and data win over our sort of domain-specific knowledge in the long term. I have some thoughts about things that make biology a little bit different. I think it's easy for a person to think that domain knowledge isn't needed to, say, build ChatGPT from millions and millions of examples of human language. But I will say people know how to look at the results of ChatGPT and tell whether it's saying nonsense. Whereas, as Yoav pointed out, with batch effects, even experts will miss when biological models are getting it really wrong, right? You can't just look at the output and tell what it's doing. So I think there's several differences here, but I will say I've seen this happen to many different fields. I certainly watched it happen in computer vision during my PhD, a dramatic shift from expert designed features to bespoke probabilistic models to neural nets. And just because something isn't working today in genomics using these neural nets does not mean that when we get 100,000, a million individuals and design our models in a little bit more careful way, that it's not going to move in the same direction as all of these other fields. I won't give you a history lesson in AI for time, but I will say I think this is interesting. I've seen it happen in other areas, and I see where we're going in genomics a bit. So it's an exciting time. So neural nets and deep learning are starting to show promise. We've had a few false starts. We haven't really had our AlexNet, computer vision moment. We haven't really had our AlphaFold moment yet, but it is exciting. I think some things to think about if you're looking at applying AI in genomics include not only how much data do we have 5 million people, but whether those samples cover the relevant axes of variation. The human genome in natural variation is highly constrained by evolution. There's a lot that we simply can't explore by looking at living people. we don't have great gold standards for a lot of the problems that we're still struggling with. Even gene networks, for example, we don't have perfect gold standards. We can create simulated data, but we don't have a perfect way to evaluate this, and we can't just look at it by eye. Like, we're not good enough at that. Machine learning, none of these methods get around confounders automatically. Some of the really exciting approaches in regulatory genomics that use convolutional neural nets, for example, specifically train their models in a way to identify confounders first and then learn the rest of the interesting stuff. I think we're going to have to think about confounders very carefully. Furthermore, if we want to come up with interesting models, not just a fully connected giant network, like what is the structure in biology and what are the invariances that we can take advantage of? So even in the applications in vision and language that are just these huge models, they still take advantage of symmetries and invariances, such as like shift invariance and rotation invariance in vision, things like that. Like, what are we going to take advantage of in biology? In addition to, of course, questions about interpretability and the ethics of applying these models in our population. So those are some thoughts I have on the AI side. So I'm going to turn away from science now and just talk a little bit about the scientific process and what these 10 years have been like. The first thing I want to show is the composition of my lab over the years. So we have run a really, really interdisciplinary lab, and I have absolutely loved that. It has been so fun to work with students and postdocs and clinical fellows across all of these different areas. And this is something that actually I'm not sure I could have done many other places than Hopkins. I interviewed at some schools that told me, yeah, sure, you can recruit students across departments, like after you have tenure, or things like that. And I've just found this to be such a collaborative environment, so interdisciplinary, so wonderful. My students and trainees like put up with me bringing this very diverse group together and sometimes like not understanding the clinical implications of what the clinical fellow is talking about, or not knowing all the biology that they know. But it's been truly wonderful to work with this diverse group. So thank you to my lab, first of all, and to the leadership for providing that opportunity. Second, I have some thoughts about team science and about how collaboration has gone. The GTEx project had some of our papers had hundreds of authors. I'm not even sure what the number was in the end. And there's criticism of how much money are we spending on these big consortium projects? So there's criticism of really what was your contribution in this big long list of authors? And I just want to say how valuable that was for my career, how valuable I think that data was and some of the results that we found from it, but also, the opportunity to be in a room with all of the experts in eQTL analysis in my first year as a professor was amazing. And yes, there was yelling and arguments. And some people didn't think we should correct for batch effects, and things like that. But we argued it out, and I think we came up with a lot of really good science, and I think it was extremely valuable for me personally. But I've also enjoyed the smaller collaborations. This is a picture of one of my former students sitting with two of Yoav's students. I can't remember if that was at Hopkins or at Chicago. They went back and forth, but these have just been really fun collaborations with experimentalists who've been willing to work together with us from the start of the experiment, not just throw us data after they've collected it, but really design how the experiment should work together. In addition to clinical faculty that I worked with, Marius was actually a clinical fellow in my lab and is now faculty at Hopkins. And he taught me more about cardiovascular disease and genetics than I ever would have known if he hadn't worked with me. And it's fantastic, and he continues to collaborate with us now. So, the collaborative environment here and just broadly how I've chosen to work with faculty and students across these different disciplines has made my job really interesting, and I've liked it. I'll make a quick comment about industry and academia. Yoav mentioned I was at Google for a long time before I went back for my PhD, not just because I hate money, also because I love genomics. But I think it's changing quite a lot. I actually looked at industry roles when I graduated. I've worked with industry since then as well. And I will say that problems that we thought, or that I thought in 2013, that could only be done in academia, they're shifting, and many of them are doable in industry now. The projects that we can take on in either place have changed, and the speed of progress has changed. So I think we really need to be paying attention to this. I mean, some of it is great. There's more opportunities for our graduates. There's more opportunities for partnership and entrepreneurship from academic researchers. But I think we also need to, I don't know. When I was at Google, I saw some research in academia become obsolete because of what was happening in industry. Industry has tremendous compute power. They have incentives that are different. And I think we need to think carefully about how we're working with them and what we're contributing that's really unique. And I'm happy to talk to anyone who's interested about that. But I think there's a lot of opportunity, but there's also some thought that is put into it. One thing I will say is that working across disciplines has kept me learning. Like, not a day goes by that I don't like learn something new from one of my students or one of my colleagues. I don't think I've been bored in 11 years. I don't know if that's good. With regard to team science, sharing credit generously, sometimes that causes some friction here and there, but it has almost always been to our benefit. It has resulted in us getting to work with experts across really different areas. Has generally, as far as I can tell, I don't know, I'm here, I got tenure, right? I don't think it's hurt us, and I don't see that it's hurt my students. So sharing credit generously has largely been positive. And thinking about the network results and a few others, I really want to think like negative results shouldn't be spurned. Some of those pieces of work that showed benchmarks or showed that previous methods weren't working how we thought, these can really excite the field. Some of those have gotten like the most notice of the work that we've done. So I don't think negative results should be ignored. I'm trying to keep this whole talk pretty positive. And last, we should keep our eye on what's happening in industry. So with that, I'll just summarize some final takeaways. And really, I showed that huge spike in data availability. Genetics and genomics has changed tremendously in 10 years, driven by data and computation. Things that were not possible when I started are now routine, and new problems are facing us. It's just been like a wild ride, and it's very exciting. Thinking about like, what can we do translationally, genetic risk prediction, disease genetics, and even personal genomics really made great strides. They are being used in clinic. They're being used in pharma. We're understanding things like time matters to genetics. We're understanding how much information we can get from other data types. So this has just been, a lot of rapid progress in these places. And I don't really think it's slowing down. I actually don't think we've extracted the full value from the data that we're gathering. Like, it's growing so fast that we actually haven't had time yet to take full advantage of it. And you can see that in things like reanalysis of public data that's been aggregated. We just published a paper today on this. There's a lot more information in there, just like GTEx led to these like 7,000 papers on everything from like mitochondrial function to loss of function to rare variants. We have not extracted the full value of the genomic data that's becoming available. I think where AI will take us is going to be really interesting to see, and how this all goes back and forth between industry, the clinic, and basic science research is going to be exciting. So with that, I want to thank my lab. The progress that we've made is your work, very simply put. You have been amazing to work with. You have put up with a pandemic. You've put up with me taking leadership roles and changing the structure of the lab and all sorts of things. And you've continued to do great work. So thank you so much to my lab. I have a bunch of, I'll keep it short, but a few more. My collaborative network, this doesn't even include students. This is only like faculty. My collaborative network has been really exceptional. I love learning new things. I love working with experts across different areas. Yoav, thank you so much for being here today and for the work we've done together. But truly, all of my collaborators have really made this work fun and exciting. In addition to the leadership, my mentors here at Hopkins in my first few years, the people I've co-taught with, my partners in leading these centers and institutes, huge thank you to all of them. Some of you are listed twice because you've done more than one thing with me, but that wasn't an accident. And finally, thank you to my friends and family, especially to my son, Clay, who was born right after I started at Hopkins and is the other reason that I haven't been bored in 10 plus years, and to my husband, Todd, who's here too. Thank you so much.